Happy National Estuaries Day! Welcome to the Grand Bay National Estuarine Research Reserve, what we lovingly in South Mississippi call the Grand Bay Near. This is a partnership in land conservation between the Mississippi Department of Marine Resources and NOAA's Office for Coastal Management. And we protect all kinds of important coastal habitats, study them, and tell the public about what they do. So today, to celebrate National Estuaries Day, we're having Seaside with a Scientist, which is a program where we've put our scientists out in the different habitats of the Grand Bay Near, and they're going to tell you about their work and what they're learning. We're happy you're here, and we hope you have a great day.
Hello everyone, my name is Marga Poston. I'm the Coastal Training Program Coordinator and the Assistant Manager here at the Grand Bay National Estuarine Research Reserve. And we're going to take you on a little tour today. So come on and let's go. So we just arrived at one of our water quality monitoring stations. The swamp coordinator, Kim Crespin, is going to talk a little bit about what we do here. She's also with Bree Andrews, our research assistant here at the reserve.
Hi, welcome. We are at the Bayou Heron Swamp Station, which is one of 4 water quality and nutrient monitoring stations that we have here at the reserve. SWAMP stands for System-Wide Monitoring Program. It's called that because each of the 29 National Estuarine Research Reserves implement this program using the same equipment and the same methodologies, so that not only can we monitor the environment in our own estuary, but we can compare that data to people from across the country. One of the core pieces of equipment that we use in our program is this oblong data logger called a data sonde, or sometimes referred to by the brand, which is YSI. This piece that I'm unscrewing is a guard because this goes under the water and we leave it for up to a month. It logs data every 15 minutes using these probes that are on it. And we don't want fish or crabs or waves even to hurt the probes, so they're covered by a guard. The things that we measure are water temperature, salinity, dissolved oxygen, turbidity, and pH. We change our data loggers every 4 weeks and sometimes every 2 weeks in summer, because when anything is in the water for an extended period of time, all of the life that is out there will start to grow on it. That's called biofouling, and it can make the probes and the body of the data logger itself very, very dirty. It will be covered in algae, barnacles, sometimes we'll get fish eggs. So we need to keep an eye on it, because if biofouling starts growing over the probes, that interferes with the readings and means that the data won't be as good.
While we're checking them and replacing them, we also bring out our YSI handheld, and that measures everything such as temperature, salinity, pH, except turbidity and depth. So this is the Secchi disc, and this is the instrument that we use to measure water clarity. We reel this into the water until we can't tell the difference between the black and the white on the disc, and that is called Secchi depth. And then we reel it all the way to the bottom, and that is called total depth.
Every research reserve in the system has at least 4 water quality sites that measure all of these things I mentioned every 15 minutes all year round, and at least 1 weather station to measure air temperature, rainfall, wind speed and direction, also every 15 minutes. We can use both of those streams of information to start figuring out how weather impacts and potential long-term changes in weather and climate are affecting our water quality as well. We have 2 data loggers for every station so that we can prepare one in the lab, have it nice and clean and ready to go, come out into the field, pull the old one, and then put the new one into the water for its 2 to 4 weeks. We download the data from the data loggers when we get back to the lab, but we also have 2 stations sending data every hour to a NOAA GOES satellite. Behind me on the station, you'll be able to see an antenna, just a silver rod with spokes coming off on multiple sides, and that's the antenna that sends the data up to NOAA's GOES satellite. So every hour you can see the latest readings. You can get those from the Centralized Data Management Office's website. There's even a version for your phone, nearsdata.org/mobile. So if you want to go fishing out on the reserve, here in Bayou Heron or near Bangs Lake and want to check the salinity or oxygen levels, you can do that in near real time. In addition to the water quality and weather measurements that are taken every 15 minutes, we go out every month and get water samples that we bring back to the lab so we can analyze them for certain nutrients. You can think of nutrients like vitamins. They are important to grow, they're important for the food chain, but if you have too much of any of them, you can get algae blooms and harmful impacts on the environment that you don't want to see. So that's another thing that we monitor with SWAMP. All of this is important not just because we're data nerds, but because we need to know what's happening in the estuary so that we can help protect it and keep it healthy for all of the critters that live there. Bree is with us today because she helps out with SWAMP a lot. And part of that is because her project works with animals that are very sensitive to water quality conditions.
So my job here at the reserve is research assistant, and my project is to test the feasibility of clam aquaculture at the estuary and along the Mississippi coast. And like Kim said, my project relies on the swamp stations, and we have 4 stations, and one of them is closer to my study site, and that's in Frutescens Bay. And we put it there because the salinity is much higher and the temperature is higher over there, but it's also prone to more biofouling. And they rely on higher salinities and higher temperatures to grow, and they're filter feeders, so the more algae in the water, the better. This is one of our clams that we had found at our shell midden recently. My clams that I study are not this large, but one can only hope that they get this big. Hopefully the biofouling isn't too bad and the clams could actually grow here and we can have a new aquaculture.
SWAMP was designed to both detect short-term variability, like you might see after a big rainfall. You would see salinity drop because you're getting all of this fresh water with no salt coming in to your system, but also long-term change. And we can look at these long-term trends to see how the things humans are doing around the waterways are potentially affecting the water itself.
This is one of our stops here at the Shell Midden and the Salt Pan, and we'll have Michael Brochard, he's our stewardship assistant. He's going to speak a little bit about the Salt Pan. And Cassie Porter, she's our wetland resource specialist, and she'll be talking about the Shell Midden.
A shell midden is basically just a big pile of shells. Historically, Native Americans would come to these areas throughout different times of the year for large feasting events, and whenever they were done eating, they would throw their shells into a big trash pile. Not only did they throw shells here, but they threw other trash items like broken ceramic glass. During archaeological excavations, they also found things like animal bones and even human remains. Throughout the Grand Bay Near, we actually have 20 different archaeological sites, and some of them can be as high as 3 feet above sea level. Another unique aspect about these habitats is the fact that they're so high up compared to the marsh around them. They can grow different types of hardwoods and shrubs and trees and some more of the stuff that you usually see upland. One of the coolest trees that's here on this mound is the prickly ash. And it's pretty cool because Native Americans were actually able to use it as a type of medicine. So anytime they would get a toothache or something like that, they weren't able to just drive up the road and go to a dentist like we can. So they had to use the plant and chew on the leaves to take away the pain from the toothache. Some of the more common trees that can be found here are things like the live oak that you can see behind me, and some of the Yopon hollies that usually just grow on the side of the woods or within the woodlands, and even some of these more tropical plants like the yuccas. So during an archaeological excavation, they would dig down to find different items that could be in the shell middens. And this is kind of what you would see if you were to participate in one of those excavations. You have just like a deep layer of shells. Some of the things that they would find are oyster shells and clam shells. And one of the oldest oyster shells that they actually found was 150 BC. Other things that they found were ceramic pottery, and there's even kind of, I guess this is a handle that they found from a ceramic pot of some sort.
Of all the interesting habitats we have here at Grand Bay Near, this is one of my favorite. What we're standing in now is the salt pan. These salt pans form whenever tidal rack and other debris washes in extreme events into these areas of the marsh and actually kill off the vegetation. If you notice over there, we've got high vegetation, and if you notice around us, it's rather barren. Those tidal situations bring in that debris, it sits down and either degrades or washes away, but when it leaves these shallow bowls. And then during successive tidal events, the water comes in, it evaporates from the sun, and it leaves behind a lot of salt on top of the soil. The soil actually can get up to 200 parts per thousand, which is, if you believe it, would be 7 times higher than seawater. That salt content really makes it hard for vegetation, like we see over there, to grow. They're not as salt tolerant, so what we have that colonize here are halophytes, or really salt tolerant plants. If you want to get down right here and look at this one, I'll show you one of the first halophytes. So this guy is called Salicornia virginica. It grows horizontally across the ground like this and roots down as it goes. If you look closely at it, it's really waxy and kind of thick, very succulent. It can hold a lot of water for this arid area right here amidst the marsh. This guy right here, actually, you can eat. Yeah.
Can I try some?
Yeah, have a bite. We call it pickleweed or picklewort. It's really salty, kind of tart. It's interesting. So we've got some other halophytes around. If you look right behind you, we've got another succulent called sea oxeye. The scientific name is Borrichia frutescens, but it is also succulent with a real thick, waxy leaf, so where it can hold a lot of water as well. It blooms a really pretty yellow flower. One of the non-succulents that can grow in this arid environment with these high salt compositions is this right here. This is saltgrass, or Distichlis spicata. The saltgrass can be identified by these spiky alternating points that go up it. Being that it's pretty barren and arid spot, you don't find a ton of wildlife in the salt pan. One thing we might find though, if you come over here and look, are these small holes. These holes are made by fiddler crabs, which really proliferate out here. They can be identified by one large claw they have that they use for both combat and to attract mates. During times of inundation, being that these are little bowls, you might get a few inches of water to a foot of water in these areas. In those times you get a multitude of fish species, fungulus and other related species. You might be familiar with the bull minnow. It can tolerate these areas that are generally a lot hotter water and really low oxygen. We've got some of the relatives of them that you'll find with them, the sheepshead minnow, the diamond killifish, and Fungulus similis, which is named because it looks almost exactly like a bull minnow, but it has a longer snout and it has a dot on its tail. And then the other thing you may find in here when there's fish in here is wading birds. So now that we've talked about the salt pan formation and what it contains, we can talk about the monitoring that we do here at Grand Bay Near. We found in some of our sites that we monitor elevation and accretion in these salt pans and in the marsh in general, that elevation change isn't keeping up with sea level rise throughout the reserve. As the water rises, we're finding that these places are inundated more and more. That little bit of extra inundation actually lowers the salinity just enough to have some of the less salt tolerant plants start colonizing these areas. If we step over here, we can see one of those plants right now. This here is Spartina patens, or another low marsh grass that is less salt tolerant. But as you can see, it's growing fine here. If you look over to your right, that sort of bluish hue grass that's low, that's Spartina spartinae. These two grasses wouldn't normally grow in the salt pan, but they are able to colonize with these more frequent flooding events. And eventually, we believe that they will reestablish into these high marsh areas. So now that you're a salt pan expert, be on the lookout for this really cool habitat the next time you're out marsh exploring. And if you're lucky, you might find a white-tailed deer out here using this place for a salt lick.
So today you guys learned about two unique habitats in the estuaries. But one of the problems with these habitats and the beauty that they provide is debris that washes in. So earlier Mike told you about tidal events and how water comes in and gets trapped. Well, along with that water comes trash and debris. And that can be really bad for the wildlife and just the aesthetic beauty of the estuary. So we have things like plastic and different aluminum cans and even fishing gear like crab traps and fishing lines. And whenever it comes here, some of that stuff can just basically get stuck here forever if we don't come and remove it.
Now we're going to look at some of the trash we picked up in the marsh. Last year during our debris cleanup, we collected over 17 tons of trash from the marsh. You can look at some of these items we've collected today and talk about the particular problems we have with those items. Here we've got bottle caps made out of plastic. We've got a plastic lid here. And that plastic can photodegrade and become small little tiny pieces of microplastics, as well as get into the digestive tracts of animals that might consume them. We've got some metal things here as well. We've got an aluminum water bottle and an aluminum can, which can break down and leach toxic metals into the environment as well. Here we've got some flat pieces of plastic, some packing material here, and some sort of wrapper that can either photodegrade down into microplastics as well, or just present the problem that they may be consumed, which could kill an animal. They may become trapped in this. They may get mistaken for prey, and just a multitude of other things. One item we have here at the Near of debris that's of particular concern is derelict fishing gear. As you can see over here, we have a derelict crab trap that was found in the marsh. During our annual derelict crab trap cleanup, we find just thousands of these things. In fact, last year we found 1,200 of these alone. These are of particular concern because they're designed to be traps. They not only break down into the materials that they're made from that are toxic and or just bad for the environment, but they can actually trap animals inside of these and kill them.
So you guys just learned about how debris can be really bad for these environments and the wildlife and just the overall look of it. So how can you help? Next time you go to the beach or go to the park, bring a trash bag with you. Make sure you clean up the trash, don't leave any behind. And if you see any trash sitting around, you can grab some of it and throw it away inside of a dumpster.
So that way we can keep this environment pristine for wildlife, for us, and for you.
Here we are at the clapper rail station. We have Mark Woodry with the Mississippi State University. He's research faculty there. And then our restoration intern, Jacob Goff.
I'm a researcher out here at Grand Bay. I'm very interested in the bird life that occurs out here on the research reserve. And one of the groups that's really a specialty for out here, because we're an estuary and we have such an extensive tidal marsh, are these birds called secretive marsh birds. They tend to hide in the grass, they're very cryptic, and they don't call very often. So we really don't know a whole lot about them, or how many are out here, or how they're important to the ecology of the estuary. Clapper rails are sort of this smallish bird that runs around in the marsh. One of their common names is actually marsh hen. They actually do look somewhat chicken-like. They're in the group of birds called the gallinaceous birds. The technical name is Galliformes, is their family name. Chickens are actually in that same bird family. So they have longish legs. They scoot around in the grass. They have a long sort of decurved bill that they actually use that bill to probe down into the marsh. And during the breeding season, during the spring and summer, they actually, about 95 to 98% of their diet is fiddler crabs. When they fly and sort of their rump is kind of hanging down, it looks like they're just really struggling to stay in the air. So they may fly 20, 30 yards at the most, but usually they kind of get tired after that. They are permanent residents here at our marshes. These birds, as best we know, do not migrate anywhere. They probably do make some local movements, like between this marsh and marshes in Alabama, but their territory size is less than an acre in size during the breeding season. They don't move very much. We've been doing these surveys since 2005. We have 50 points scattered across the reserve, and we monitor those every year to try to understand how populations of clapper rails and other marsh birds change over time, but then also what habitats within the marsh do they prefer, if they have a preference at all. We're actually going to do a shortened version of an actual survey for you all this morning. But it basically consists of, we stand on the bow of the boat, we listen for a 5-minute period without any calls or anything, and we record any of the birds that we hear. And then we actually have a speaker that Jacob is holding, and we have different marsh bird species calls recorded. He'll play those calls, he'll broadcast those calls out into the marsh, and then the idea is that because these birds are territorial, they will actually call back. So what we're going to do is we're going to take a minute. We're not going to make you all sit here and listen for 5 minutes. We're going to take 1 minute, and we're all just going to listen passively and see how many birds we detect. Jacob and I will be sure to let everybody know if we hear something. We'll kind of point. And then after we do that, then we'll actually use the call broadcast and try to see if we can get some more birds to respond. So if you all are ready. One clapper rail just called kind of back here behind us, probably 50 yards away or so. That's pretty awesome. Okay, so that was a minute. So we had one individual in that 1 minute call. So what I'm going to do now is have Jacob is going to play a series of clapper rail calls. And then once he plays all those, then we'll actually start listening for a minute and see how many birds we pick up after that. Clapper rails have a couple of different sort of common calls they use. One is called a keck, where they just, and this is the first one you'll hear, they're just kind of like keck, 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 keck. And that's just sort of like a contact note letting them know like where their mate is, stuff like that. Then you'll hear a call called a kekara. It's like kekara. And that's sort of a male sort of call of like, hey, I'm over here, and kind of, stay out of my territory kind of thing. And then you'll hear a call called a kekbur. It's like kekbrr, kekbrr. That's a call that females actually give when they're ready to mate. And so that call is often one that actually causes most of the clapper rails to call back to us. So like I say, Jacob will play a series of those calls. We'll kind of just listen, and then hopefully we'll hear some more clapper rails than we did in the passive period. So. Yeah, just kind of play them all the way through. So now there's one calling behind us. So that's a new one from the one that was over there. Here. So we've got one back here and one over there. So those were the kek call, those are the contact calls. This one's calling back here again. That was I missed one of the calls. That was the clatter or the clapper call. So it kind of sounds almost like people are clapping their hands. So that's where the name clapper rail comes from.
That was clatter.
Yeah. And that's the kek hurrah call. That's like the male being a little agitated and sort of just letting everybody know he's not happy. I think back behind us there's one actually not too far across the bayou, and then there's one farther out.
There's an alligator crossing behind you, Mario.
So that's the kek burr call. Run through them again, Jacob.
Kek burr and clatter.
Yeah.
Right there.
There's another one right across the way, so that's 4 so far versus the 1 that we had in the passive period. So we'll listen here for a few more seconds and just see if anything else calls, and then we'll summarize the results from our one calling from right here,
Maybe 30 feet. Sorry, you're fine. You're fine. So, play the kek burr call.
So that was 5 birds that we've had versus the one we had before. So obviously we would be underestimating the number of birds around. What we're going to do is we're going to play a couple of calls to try to get this bird to come out, to get a little bit closer. And I'll explain to you why this bird's coming closer here after we play a few calls. Yeah, and now there's 2 more calling way, way out. So that's a total of 7 that we've detected at this point. One of the ideas behind this call broadcast survey, and the reason that it works fairly well, is that these birds are territorial. And so by using these call broadcast surveys, a bird that may have a territory in this portion of the marsh actually hears this call broadcast and thinks that it's actually a competitor that's trying to move in on its territory. And so just like people start messing around on your yard or something like that, you're going to go just kind of ask, hey, what's going on? Why are you, in my yard, whatever. That's the same sort of thing that's going on here. Basically, these birds are moving in. You got to try to see where is that clapper rail and who is he and why are they here and, what are they up to? So that's the basic premise behind using this call broadcast. They're probably the size of a football. They're sort of brownish and reddish and gray. So if you look out into this marsh vegetation, you could see that the bird could literally be right in front of you, just like that one's calling literally probably 5 or 6 feet from me. And there's just no chance of seeing it. One, because the vegetation is so thick, but also because they're so cryptically colored. The other thing about clapper rails is it looks like, you've all probably heard the phrase skinny as a rail. That phrase comes from these birds called clapper rails, king rails, and several other species of rails. And the reason they use that phrase is if you had a football in your hands and the ends of the football pointing away from you and your hands on the side, if you took and just squeezed the football in, and it would be really narrow, long and narrow, that's how clapper rail bodies are shaped. And that's more than likely an adaptation for making their way through all of this really thick marsh grass. They're very cryptically colored. Obviously they don't call a lot, but they will call if they're provoked or if they think there's an intruder in their territory. Clapper rails, like most birds, are territorial during the breeding season. Once we get to later in August into September, those territories tend to break down. The birds seem to spread out more evenly across the marsh. Typically, when we do these kind of surveys, we'll only get 1 or 2 individuals at the most during that passive period. But then when we start playing different marsh bird species calls, you should be able to hear that a lot more marsh birds will actually respond. And so that allows us to actually get a better estimate of how many marsh birds are actually out here. If we only use periods where we came out and just listened passively without any call broadcast, we would way underestimate the number of clapper rails out here. If we're trying to understand how these populations respond to things like non-point source pollution or hurricanes or fires that may happen in the marshes or something like that, we really want to have a better idea of how what the population numbers are like. This is a standardized technique that's used across the entire country. Most of the techniques we use to study their movements are based on radio transmitters, these little transmitters that we put looks like a little backpack of monofilament line that we put on them, and then we can track their movements both by hand, and then we also have these automated recording systems. The problem is those transmitters, the batteries only last about 3 months. So if we put them on in March or April, by the end of the summer, those batteries are dead. The birds are much harder to catch once they're outside the breeding season because they don't respond to call playback as much. So there are some new technologies out there that we're probably in the next year or so going to be testing some new transmitters that are solar powered, so they should work indefinitely. They'll send a signal to a satellite, and then we just go to a website and we'll download those locations from a website. But it'll actually give us the location of that bird within 2 meters on the ground. It will tell us where that bird is. That's all we got. I appreciate it, and we'll see y'all around.
Here at the Critter Station, you'll find Andrew Heaton. He is the Restoration Monitoring Specialist, and with him is Mike Archer. He's our coastal ecologist. And we also have Jennifer Cumbus. She's the Education Specialist.
So we have a two-toed amphiuma here. So these guys look a lot like snakes, but they're actually aquatic salamanders. And if you look closely, you can see two pairs of tiny limbs that they have.
All right, so this guy right here actually has a few different names. You may know him as a crayfish, a crawfish, or even a mud bug. And all of those are based on where in the country you are and how you like to eat them. So they're really popular for humans to eat, but one of the really cool things about them is they belong to a family called crustaceans. And they are called that because they have this really cool exoskeleton. So you and I, we have an endoskeleton. That means all of our bones are on the inside of our bodies. This guy, all his structure, the things that allow him to move and the stuff that protects him is actually found on the outside. And that's why we call it an exoskeleton.
Our next critters are fiddler crabs. Fiddler crabs are found all over the marshes and in habitats like salt pans. They are pretty cool. They're sexually dimorphic, meaning that the males and females look a little bit different from each other. The males have a huge claw that they'll use to attract females. They're also delicious to animals that live in the marsh, like diamondback terrapins and clapper rails and all other sorts of creatures.
These right here are hermit crabs, and there are 800 different species, so that's quite a lot. And you can see that they have this shell. So all their bodies at the very back have this nice hook structure, and that actually allows them to stay inside of that shell. And as they grow, they will find bigger shells for them to have as their home. And what's really cool is once a hermit crab dies and vacates its shell, it actually releases a chemical that tells all the other crabs in the area, hey, my shell is now available. So anybody looking for a new home can be able to find one.
Alright, so this guy here is a Mississippi green water snake. So even though a lot of people think these guys are dangerous, they're completely harmless. They're not venomous like cottonmouths. You can tell them apart by them not having the triangular heads that cottonmouths have, even though they live in the same wet habitats. So this guy here is a squirrel tree frog. So it looks a lot like a lot of the green frogs you'll have hanging out outside your house, but these guys actually get their name because when they're calling and talking to each other, it sounds a lot like squirrels chattering.
Here we have a stone crab. It gets its name from looking like a stone underwater when it folds its arms in. You can tell the male and female apart because of the pattern on the base. This is a male because of this lighthouse or Washington Monument shape. A female will have more of a U shape. Blue crabs have similar patterns.
Alright guys, this is a diamondback terrapin, and they are recognizable by this really unique, really pretty skin. So if you look, it's kind of this grayish color, and it actually has these really pretty spots. So that's one of the main reasons we can tell it's a diamondback terrapin. Another is the scoops on its back are actually in a diamond shape, which is another way that it gets its name. And we can tell that this guy right here is a male because he's actually smaller. So the females will get a lot bigger. And he also has this really long tail. And so the females will actually have much shorter tails than this as well. And if you notice, there's this nice big contraption next to me. And this contraption is called a crab trap. So what people do is they drop them in the water and they will try to catch crabs. Okay, crabs can be pretty good eating, so you know, can't really blame them there. The problem is that this guy right here loves to eat crabs just as much as humans do. And so he actually gets stuck in these, and because he needs air to breathe, when he gets stuck in these, it ends up harming him. He ends up dying. And so we are working with crabbers to use what we call turtle exclusion devices, also known as TEDs. And they are these orange squares that you can see in this crab trap here. And what that does is because it is a rectangle in shape, and because this diamondback terrapin is dome shaped, when he tries to go in that crab trap, he can't get in. But the crabs, which are flatter across the top, can get in. So the crabbers are happy because they still get to eat all their yummy crabs, and we are happy because our beautiful diamondback terrapins stay nice and safe.
Here we have a yellow garden spider. It's an orb-weaving arachnid. Typical spiders have 2 claws on each foot, This one actually has 3 to help it spin its complex webs.
Hey y'all, I hope you had an amazing experience. I hope you learned something new about the amazing coastal habitats of South Mississippi and got excited about science. Now it's time for you to go out and find your local estuary and find something to do to celebrate National Estuaries Day.
Happy Estuaries Day!
Happy Estuaries Day!
Happy Estuaries Day!
Happy Estuaries Day!